Bismillahirrahmanirrahim. Today we will talk about neurodegenerative disorders, and in this we will be discussing the definition, classification mainly of Alzheimer's disease and Parkinson's disease, and also the pathogenesis, morphological features, clinical correlation and complications, and then finally diagnosis. Now, what are neurodegenerative disorders? There is cellular degeneration and progressive loss of subsets of neurons and their interconnections. Underlying the pathology is the abnormal accumulation of certain proteins which damage the neurons and their synapses. This abnormal accumulation of proteins actually results because of certain mutations which may result in the imbalance between formation and degradation of such proteins. Another important thing is that these abnormal proteins don't stick to one part of the nervous system, but they tend to spread and involve other normal neurons. And this accumulation or aggregation of abnormal proteins can result in the activation of immune response and triggering inflammation and causing further damage. Now, if we classify neurodegenerative disorders, this may be in the cerebral cortex and hippocampus. In this case, the most important being Alzheimer's disease, which is basically initially to begin with cognitive dysfunction and leading to later on severe dementia. Then we have basal ganglia diseases, which result in movement disorders. One being causing hyperkinetic, others hypokinetic disorders. Parkinson's disease is the prototype of hypokinetic movement disorders. This results because of the degeneration of dopamine secreting neurons in substantia nigra. Then is the Huntington's disease, which is a prototype of hyperkinetic movement disorder, and here it is mostly autosomal dominant and results in the damage to the caudate and putamen. It is characterized by jerky movements throughout the body and the peripheral extremities, especially in the upper arms, chorioarthroid writhing movements. There is also accompanying cognitive impairment and later on severe dementia. This because of the trinucleotide repeat mutation of CAG on chromosome 4, which results in the accumulation of abnormal Huntington's and ubiquitin proteins in the neurons, causing damage to the neurons and their synaptics. Then there are disorders of cerebellum and spinal cord because of the degeneration mainly in the spinal cord, cerebellum, and the peripheral nerves, mainly the sensory nerves, and resulting in movement disorder causing sinocerebellar ataxia. So there will be cerebellar disorder with sensory ataxia, rigidity, sensoromotor peripheral neuropathy. Again, this is caused by CAG repeat expansions resulting in accumulation of abnormal proteins which cause damage to the nerves. Finally, upper and lower motor neuron involvement. And the typical disease is amyotrophic lateral sclerosis. There is involvement of both lower motor neuron and the upper motor neurons. Lower motor neuron results in the weakness, muscle atrophy, difficulty in movement and fasciculation, whereas the upper motor neuron, as you know, may result in spasticity, hyperreflexia, and paresis. Later on, there is disturbance in swallowing, breathing difficulty, and this may result in aspiration, pneumonia, and death. The underlying pathology in familial type is the mutation in the SOD1 gene, which is present on the chromosome 21, and this results in the accumulation of SOD1 protein, and sometimes on chromosome 9 there is mutation and result in hexanucleotide repeat mutations. We'll be talking in detail Alzheimer's disease and Parkinson's disease. So as already mentioned, one of the neurodegenerative disorders, and it's in fact the most common cause of dementia. Usually occurs above the age of 50 years, and with advancing age the incidence increases. To begin with, mainly it's a cognitive dysfunction characterized by memory loss, which results later on in severe dementia. There may be mood variations, behavioral disturbances, and so on. There is abnormal accumulation of A beta protein and also tau protein in the form of plaques and neurofibrillary tangles respectively. There are two types: sporadic, which is more common, and familial, which occurs early, about 5 to 10% of the cases. There is mutation in APP gene. APP is amyloid precursor protein gene, and APOE gene on chromosome 21 and 19 respectively. And the genes involved are in fact, presenilin gene on chromosome 14 and presenilin gene on chromosome 1, which is concerned with the familial type of this. Now, it is more frequently seen in Down syndrome because of the trisomy 21, because there is extra copy of chromosome 21. As far as the pathogenesis is concerned, there is increased formation of beta amyloid protein from amyloid precursor protein. And this is because of the mutation in APP gene that encodes the amyloid precursor protein. Now, two enzymes are involved: base, that is the amyloid converting enzyme, and secretases, in which most important being gamma secretase. And this results in abnormal cleavage of the segment which is toxic, and this aggregates and results in the neurotoxicity. The sporadic cases are as a result of lipoprotein E mutation. With the accumulation of A beta amyloid protein, there is decreased clearance and results in neurotoxicity. Now, if you see this diagram, normally this APP molecule is cleaved by the enzymes base and gamma secretase and also alpha secretase. But with mutation in these genes, there is abnormal excessive cutting at this place, and results in the formation of plaques known as beta amyloid plaques, and they have the tendency to accumulate and as oligomers and then as filaments, and they are difficult to be cleared. Now, if you see this segment and this segment also, when there is cleavage by beta secretase or base enzyme, this results in the formation of insoluble aggregates into plaques known as A beta 42. It is in fact this segment which is neurotoxic. Other abnormal protein which is accumulated is tau protein. Now, tau protein is normally present, and it is associated with microtubules, which actually give stability to the axons. Now, as a result of the mutation, there is abnormal accumulation of hyperphosphorylated tau protein, and they also change their position and results in the occurrence of neurofibrillary tangles in the cytoplasm of the neuron. So it is, to begin with, intracellular structure, and later on, because of the neuronal death, it becomes extracellular. So there is increased aggregation of insoluble, this misfolded protein. This is a helical structure, and that results in the reduction in the cholinergic activity of the cerebral cortex. Now you see normally this tau protein gives stability to the microtubules surrounding the axons. When they become hyperphosphorylated, they change their position and they accumulate in different positions as tangles, and this results in the instability of the microtubules and later on axonal damage and neuronal damage. Now if you see the morphology, this accumulated as plaques known as neuritic plaques or A-beta amyloid plaques. In the center there is accumulation of this A-beta amyloid surrounded by dystrophic neurosis, and this structure is found in different parts of the cerebral cortex, mainly in the gray matter. It measures up to 200 micrometer. It is already mentioned surrounded by presynaptic neuronal processes with reactive gliosis over here, and this may also be found in other areas like amygdala, hippocampus, apart from cerebral cortex. Now the morphology of tau protein, they accumulate as neurofibrillary tangles. In fact, these structures are double helical structures like this, just like corkscrew. If you have seen corkscrew, it has a structure which you can hold, and then the structure which can pierce the cork, and you can actually open the bottle by removing the cork. So this is a double helical structure, and this is formed because of the hyperphosphorylation of the tau proteins. Now mostly it is seen in hippocampus, amygdala, cerebral cortex and subcortical gray matter, and it can be stained by silver staining as tortuous helical corkscrew-like structures 10 nanometer in diameter. Naturally you will see them like this. Now there is another protein, another structure which is involved by this amyloid material, that is in the vessel wall of arterioles and capillaries of the brain. And like this is the accumulation of A-beta amyloid in the vessel wall. This can be seen in the otherwise in the old age, but is typically seen in Alzheimer's disease. This results in severe cortical dysfunction, and mainly because of the A-beta amyloid plaques, and also there is role of neurofibrillary tangles containing hyperphosphorylated tau proteins. In the early stage, there may be short-term memory loss, but later on, severe cognitive dysfunction with mood variation, behavioral changes, language orientation, and attention affected. Later on, complete dementia. To begin with, it is the cognitive dysfunction, but later on it is leading to movement disorder with disabled and immobile individuals, and death usually occurs because of the aspiration pneumonia and intercurrent infection. Now, as far as the inflammatory response is concerned, this is triggered by A-beta proteins, and these are actually the response is because of the fact that the inflammation is triggered to clear these A-beta proteins, and because of the proliferation of microglia and astrocytes. But during this proliferation, there is liberation of certain cytokines which also damage the neuronal injury. So there is a secondary damage to the neurons as well. Now, clinical correlation is there, but the laboratory diagnosis as such, we can do the CT scan to detect plaques and cerebral atrophy, and also you see, if you see this area, this is not taking the dye, and this will, you can just temporal and parietal areas because of the cerebral atrophy. And also there is dilatation of the ventricles because the gyri are atrophied and the sulci they are widened. Presence of neurofibrillary tangles and amyloid plaques using staining techniques can also be seen in autopsy findings in silver staining. Now let's talk about other important disease, Parkinson's disease, which is concerned with movement disorder, and this is referred to as primary or classical Parkinson's disease. It's basically a progressive neurodegenerative disease associated with damage to the dopaminergic neurons in parts of the brain, mainly nigrostriatal pathway, that is the fibers projecting from the substantia nigra to the striatum in the basal ganglia. So it is a typical example of hypokinetic movement disorders, any decreased activity and decreased movement disorders. Prevalence is 120 per 100,000 with slight male preponderance. The incidence is increasing with advancing age, and especially more so above the age of 60 years. Now, what is Parkinsonism? Parkinsonism is actually a clinical syndrome which is characterized by diminished facial expression or marked face, postural instability, stooped gait, slowness of voluntary movements, fascinating gait, rigidity and spasticity throughout the body, and appearance of involuntary movements. So this is a symptom complex. Parkinson's disease is part of Parkinsonism, and this mainly seen because of the damage to the dopaminergic fibers in the substantia nigra. So Parkinson's disease may be sporadic or familial, which is most common, but other causes of Parkinsonism include cerebral infections like encephalitis or ischemic damage or carbon monoxide poisoning, phenothiazine as antipsychotic drugs, certain pesticides, then brain tumors, metabolic disorders, chronic liver disease, and hypoparathyroidism. They're all part of the Parkinsonism, which is a symptom complex. And Parkinson's disease is included in Parkinsonism. Now we'll talk in detail about Parkinson's disease. So this disease is associated with paucity of voluntary movements, increased involuntary movements as a result of accumulation of abnormal protein known as alpha-synuclein. There is also mitochondrial dysfunction and abnormalities, and mainly occurring because of the neural damage to the substantia nigra. Now, if you see the clinical features, you remember TRAB, T-R-A-B. T stands for tremors, which are resting tremors. R for rigidity in the muscles of the body. A kinesia, difficulty initiating movement, and bradykinesia, slowness in the execution of movements. Now, the genetics of Parkinson's disease, basically there is accumulation of alpha-synuclein protein, which is actually normally present, concerned with synaptic transmission. But here there is abnormal accumulation of this gene, protein, which may be because of the autosomal dominant or autosomal recessive pattern of inheritance. There is decreased clearance of this protein because of the defect in autophagy. Now, genes involved are alpha-synuclein gene and LRRL2 gene. And in autosomal condition, there may be involvement of Parkin gene. Mainly, there is defective clearance of this accumulation of abnormal protein known as alpha-synuclein. So, when this alpha-synuclein aggregation is there, because of the decreased clearance by autophagy, there is also altered mitochondrial dysfunction and results in damage to the nigrostriatal pathway, and there is degeneration of neurons as a result of this accumulation of proteins. Now, normally there is a fine balance between the formation of dopamine and acetylcholine. In fact, dopamine has an inhibitory influence on acetylcholine, and this balance regulates the posture, muscle tones, and voluntary movements. Now, if there is an imbalance, there is excessive cholinergic activity, and this may result in the excitatory acetylcholine causing most of the dysfunction. Now, as far as the morphology is concerned, there are two important things. There's pallor of substantia nigra. You see over here, this chordate nucleus with the dopaminergic fibers which are pigmented, they are lost. This is the normal one, and you see the pallor over here. And then formation of accumulation of the Lewy bodies and neuritis. Lewy bodies are single or multiple tortuous eosinophilic bodies surrounded by dystrophic neuritis. You can see over here as well. These bodies consist of filaments composed of alpha-synuclein and ubiquitous. And dystrophic neuritis also contains alpha-synuclein. Now the clinical features. I've already mentioned that it's a movement disorder. The slowness or paucity of movements. There is, in fact, throughout this rigidity in the muscles of the body, which it keeps on increasing as the disease advances. In fact, mostly it's a clinical diagnosis. The cardinal signs being movement disorder, there is postural instability, stooped posture, and rigidity throughout the body, difficulty in initiating movement, and then slowness of movement. A very important sign is clasp knife rigidity. Now, if you close the fingers and try to open it, there will be a jerky resistance. Like when you open a clasp knife, it opens with jerk. So there will be jerky movement while opening the fingers. And the involuntary movements, like pill rolling movement in thumb and index finger. If you see the diagram, this expressionless face or mask-like face is stooped posture, rigidity, and usually there is some flexion at the wrist joint and forward tilt of the trunk. Reduced swinging of the arms, slightly flexed hips and knees. And the gait is very important to note. When the person tries to walk, it is difficult to initiate the walking movement. And initially, the person takes short steps, known as shuffling short steps. And later on, there is acceleration of this short steps, and this is known as fascinating movement. So there will be postural instability as well, and the patient may fall. And later on, there may be cognitive disturbance in cerebral cortex resulting in dementia. So this ends with our today's topic, and this will be uploaded as a form of PDF and also as a recorded lecture. Thank you very much.